Bom, pessoal, boa tarde a todos. Muito obrigado uma vez mais por estar aqui presente num novo CEBIMário. Então, hoje teremos de convidada a doutora Adriana Alzate, que vai nos apresentar um pouco sobre dispersão e distribuição de espécies. Agradeço a todos que estão aqui já presentes para esse novo CEBIMário. E a gente vai meio que mudar de idiomas. Lembrem que sempre as perguntas podem ser feitas em português ou em inglês, em caso vocês desejem fazer. E agora, então, vou apresentar a doutora Adriana, que está aqui com a gente hoje. Entonces, Adriana, muchas gracias por aceptar participar de
Gracias
esto.
por la invitación.
Es muy chévere, después de muchos años de haber salido del mismo laboratorio en Colombia, vernos ahorita en este espacio y conocer un poco más de tu trabajo. Y bueno, entonces ahorita voy a volver a cambiar a portugués para presentarte y luego te paso la palabra en inglês. Bueno.
Ok.
Então, pessoal, a Adriana, como eu acabei de mencionar com ela, a gente saiu do mesmo laboratório na Colômbia com o nosso ex-supervisor Fernando Sapato, e agora ela fez mestrado, doutorado na Holanda, e agora ela está atualmente vinculada a um pós-doc no iDiv, na Alemanha. Então, ela vai falar um pouco mais do trabalho que ela está desenvolvendo e das coisas que ela está fazendo nessa palestra aqui. Então, thanks, Adriana, for accepting this participation in this talk. I'm very excited about listening more about your work. And okay, the room is all you.
Okay, thanks, Juan Paula. Thanks for the invitation. I'm happy to be here. The first time I actually talked to any lab in Brazil. Very cool. So, as he said, as Juan Pablo said, I am a postdoctoral researcher in iDiv in Germany. And today I want to show the results of my study on understanding how dispersal affects range size. This has been a work that has been together done with Renske Onstein from iDiv and is for the moment provisionally accepted for ecology letters. So first of all, I guess many of us has asked this at least once in our life at times, like why some species are more widespread than other species. Like for instance, you see the common barn owl occurring in several continents, in most of the continents, or the ocean sunfish occurring in most of the seas. Whereas another species, and here just I have an example of the Iberian lynx occurring very narrowly places in the Iberian Peninsula. Of course, there are several ecological and evolutionary explanations of why differences in distribution of species, and they can range from differences in environmental tolerance, differences in colonization of extinction dynamics, also differences, for instance, in trophic level or the species age, like older species might have more time for range expansion than younger species, and of course dispersal, and many other ones. And all these factors, of course, act together simultaneously to determine the current range of a species. But of all of this, dispersal has been the one that has received quite the most attention. And why is this? It's because dispersal is actually a very prominent process that affects range sizes. And this is because it can affect the mechanisms for range expansion. And in this case, if you see, assuming that the gray area is the current range of a species, it can promote colonization of new habitats. But in addition, these new habitats that, of course, the species might be not very well adapted to them, it can also prevent extinction by demographic rescue, bringing individuals of the time some populations don't go extinct. But in addition, it can also provide genetic rescue by bringing sometimes some genetic variants that allows the population to locally adapt, and therefore they can allow to expand the ranges. Of course, dispersal has two kind of twofold effects on adaptation. It can also, if there is too much dispersal, it might bring a lot of maladapted individuals, and by gene load or gene swamping, then you can just actually have maladaptation and the ranges might not expand. In addition, theoretical work has been showing that dispersal actually affects positively range size. And here there are two examples using spatially explicit neutral models. For the ones that are not very familiar to this, these models assume that all individuals in a meta community have similar or equivalent characteristics. For instance, they have the same rates of probabilities of dying or of giving birth, of dispersing. And by modifying these parameters, especially the dispersal parameter, you can just predict what will be the distribution of ranges through time. And in the left example, then you see there are panels with numbers from 1 to 4. Then when you increase, and this is an increase in dispersal distance, the more, this is a distribution, a frequency distribution of ranges, that the more dispersal, the higher the dispersal distances, the ranges are more shifted to the right. That means larger ranges. And also, and in the right panel, there is another example that I have been doing also with neutral models, trying to explain the distribution of ranges in the tropical East Pacific for reef fishes. And here is kind of showing similarly like the frequency distribution of ranges, but in a little bit different way. This is like inverse cumulative distribution, which means if you see the x-axis is the relative range, which 100% is the maximum range any species can attain, in this case in the region. And in the y-axis is the number of species that have ranges that are larger. That's that certain point in the x-axis. And then we see that they have panels. The top panels are for fishes that with no pelagic eggs. Those are fishes that have less dispersal capacities because they have two ways of spawning the eggs, either pelagic or no pelagic. And you can imagine that pelagic eggs have more time to spread passively through currents and therefore have larger dispersal capacities. And also how mobile the adults are. So just to see two examples, in the bottom left, this is the kind of the guild for the species that have the highest dispersal capacities. And then you see, I don't know if you see my cursor. Let's say if you just check in the x-axis in 80, and then you go to just to see what, you see that most of the species have ranges that are large, like larger than 80% of the complete range. And the complete contrary example is this one to the top right, that is for species with no pelagic eggs and low adult mobility. Just let's assume like damsel fishes with like more territorial behavior, that they don't move more than a couple of meters during the whole lives as an adult. And then you see that here, very few species have ranges that are larger than 80%, for instance, of the ranges. So there are very few, the ranges tend to be small. And of course, there is a lot of variation, but in general, there is this tendency that with an increase of dispersal, many of the species have larger ranges in comparison to the species that have groups of species that have less dispersal capacities. But this is, of course, theory. And what the empirical data shows? And here is just an examples of studies that have examining this in different clades. So you see beetles, insects, we see plants, we see birds, fishes, and you see that there is a complete mixed outcomes. So some studies show positive effects of dispersal on range size, whereas other ones show neutral, means no effect, and other studies have shown mixed results. Of course, this has led to the idea that maybe dispersal is not that important, maybe other factors might be more related to increasing range in the species. So of course it came the idea for review, and it was to really check for all the studies that have examining these questions and really investigate whether dispersal actually has an effect on range size, whether this is positive, and also to understand why is this mixed outcomes. So the first thing that comes to my mind, and hopefully to your mind, is that all these studies differ in several ways. So there are biological differences, like all studies, even for the same taxonomical group, use different dispersal proxies. So then you can use, let's talk about fishes, then you can use the type of egg or how long the larvae can last in the water column. Or for birds, you can use body size, but you can use, for instance, hand wind index. And there are many proxies that people use, and you can maybe think that some proxies might be better than other ones, so might capture the dispersal process a little bit different. Also, the studies might examine endotherms and ectotherms, and these things might be might have, well, an important difference. The studies also differ in some biogeographical aspects. There are different realms, so there are studies in freshwater, studies in terrestrial systems, studies in marine systems. This might be causing a complete different outcome because this is assumed that, for instance, in marine systems, that the dispersal is easier because of the nature of the dispersal medium, the water, so that allows easily for passive dispersal. So if everybody can reach everywhere, so you will expect that the relationship between dispersal and range size might not be evident, or it might not be actually exist. Also, the studies might have been done in different latitudes, and there might be also difference there because the tropics might have been constant with environmentally constant through time more than temperate regions. Also, studies study different regions, so you might expect that what happened in Australia might be completely different to what happened in Africa or Europe. Evolutionary differences, so studies examine different clades. They might consider the age of the species, or they might not consider that and study all these with the species with different ages that might have some confounding effect. They might consider phylogenetic relationship between species. And of course, there are methodological differences. For instance, how you measure the range. And the range, in general, you can measure it as extent of area of occupancy or an extent of occurrence. And within these two groups, there are many different ways to measure it. Also, how complete the people measure the range. Is this the complete range, or is it the range within a continent or within a country? Which is how many species you use to study these questions. And it ranges from 5 species to 10,000 species in some studies, like using birds. And the taxonomic unit. Most of the people uses species level as the taxonomic unit that you just want to investigate, whereas other ones use family levels or genus levels. And the taxonomic breadth that is also interesting is because this means, well, what is the taxonomical level that groups all the species in your study? And for instance, some studies examine all the species within a genus, where other studies examine several species within a kingdom, which happens more often in studies on plants. Okay, then how did we do that? What we did was a systematic review and a meta-analysis. For the ones that don't have much idea what is this, is that you go to, for instance, Web of Science, you just put some key terms that describe your question, like I wanted just to put key terms that will lead me to a study that examine dispersals and range size, and then you just make a query, and then you get all the outcomes from this engine. So we end up with a lot of studies, like 3,000 studies, and from those ones you have to screen and check which ones are the ones that actually work. And the funny thing is that most of them were out because they were not even related to biology, math, physics. For a reason, ranges and dispersal have something to do with physics. And we end up with 104 studies that you have to go over and read them completely and really check if actually is what you need. And I excluded some studies that were, for instance, of range expansion because those are based on one species, or only studies, like theoretical studies. And finally, we end up with only 86 studies that were adequate for this review, and in total 575 relationships between dispersal and range size. Of course, you see that big number in relationships is because one single study can examine the same thing for different groups or in different ways. So this is why we have sometimes several relationships within a single study. And we use these ones to do more of a descriptive or more like a review, or more descriptive, or what is also called vote counting, and to do the proper meta-analysis, what I call the hardcore meta-analysis. You have to actually, from these studies and from this relationship, get actual data, which means effect sizes, correlation coefficients, and some information on the sampling variance. And this is basically, I just use it mostly for this sample size, like how many species you actually use. Because this one is very important, because it will give you more weight when you examine this. It will give more weight to studies with more precision, which means less, like a smaller sampling variance or more species, than studies that have not very precise. So what we define, what actually the data shows, and this is based on vote counting. So from the 86 studies and 575 relationships, and what I mean with vote counting is that you go to studies, you see what they report based on p-values, and of course then you can just know which studies are positive, which ones are not significant, neutral, and which ones are negative. And we see that in the A panel left, that most of the studies actually show neutral relationships. That is like around 56%. A large proportion shows positive relationships, and very few show also negative relationships. You see, it's a very big mix of outcomes. And this also happens not only like for along the whole tree of life, from plants to all, but several organisms, marine organisms, but you also see that in taxa like mycorrhiza, liverworts, diatoms, trematodes. Well, you see all these mixed outcomes. So this is why it comes in handy to do the meta-analysis, because then you have all these things of positive, neutral, but you really want to know exactly what is the effect of dispersal. Then this is why we did the meta-analysis. And what did we find? We actually found that overall, the effect of dispersal, by including all the sites together and all their effect sizes that are weighted by their sampling variance, that overall dispersal has a positive effect on range size. And also we examine what if there are differences between realms, and we saw that there are no differences between realms. Actually, marine systems do not seem to have marine systems don't seem to have like less positive relationships than terrestrial systems. But freshwater systems, which are, you see this humongous variance, is because, then you see what happened with freshwater systems, is that there is not much data on freshwater systems. Then you see the freshwater system, the gray kind of circles. So there are in total 7 studies. 2 of them have been global, mostly with fish, and there are only 5 studies which have been done in the northern latitude. So with freshwater, we cannot we draw very strong conclusions. We also examine whether there are differences among clades for the relationship between what is the effect of dispersal on range size. And for that, we did a phylogenetic meta-analysis that is just to consider the phylogenetic relationships between groups. In this case, was around the order level. It's impossible to do a meta-analysis including each little relationship, because we don't have species level data, but it's mostly a community meta-community level data. So this is what has been done at the order level. And then what we found is that there is not a phylogenetic signal of the effect of dispersal on range size. This is what we see here in the left, this sigma 2. And also it shows, with the test of moderators, it shows what is the effect of clade on the effect of dispersal of range. And we see there is not effect of clade on how dispersal affects range size. So we conclude that the clade per se is not something that will affect how dispersal affects range size. But something that is also was very clear is that, well, as I told before, is that all the studies, all the studies use a different dispersal proxies, even within the same taxonomic group. You can choose your favorite taxonomic group to the right and see which traits people uses. So we see that different dispersal proxies actually show different dispersal range size relationships, and it's quite mixed. This is based on both content, that's p-values. So green ones is positive relationships, the red one's negative, and the yellowish is neutral. And then you see a whole mix of things. And even for each taxonomic group, you see a lot of mix. And even for the same type of trait for different taxonomic groups, you also see a lot of mixed outcomes. And something that is key here is what we think dispersal is, and how we define dispersal, and how we think define dispersal proxies. And this is something critical, because actually there are many definitions of dispersal. Here I just put a set of them. I will just read, well, 1, 2. For instance, this is one of the most common ones from Ophelia Rolls. Dispersal is any movement of individuals or propagules with potential consequences for gene flow across space. And this is just to be clear, potential consequences. And there is another one that is also, I like it a lot, is that dispersal is an outcome or an emergent property of a multi-stage process that includes departure, transfer, and settlement. And from all the definition, it's very clear that dispersal is something that is complex, that dispersal might lead to gene flow, and there is something that is actually that emerge, and it's an emergent property, because you cannot really measure dispersal as a number, or it's not a single trait that really determine what dispersal is, but it's just the outcome of several traits acting together simultaneously or at different stages in the lifetime of an individual. And then, of course, it comes what are dispersal and what are dispersal traits. So from before, I said that dispersal has 3 different stages, and we are most used to one of the stages that is the transfer phase, that is mostly the movement from the natal place to a new place. And of course, for this one, we have different traits they can be morphological, for instance, the size of the wings of the birds, this kind of traits that we are more used to it. But there are other phases, for instance, the departure part, that is when the individual in a place decide to leave this place. And that the probability of leaving your natal place, it depends on several things, and it could be the social context, or for instance, there is a lot of competition, like king competition around, so they decide just to leave. There is an interesting example. Then you see this in one of the pictures in the top left. Those are spider mites. Those are some acari that they have this crazy exploitative behavior, that they just go to a plant, they consume everything. When everything is gone, the females decide to lift their little legs and let the wind bring them somewhere else and disperse. The rest of them, they stay in the sinking boat, of course. Well, and then we have the last stage after the transfer, that is settlement. You have to arrive to a place, and then you have to actually to successfully colonize it. And of course, there are other traits that are important for it. Let's think about plants. If you are a plant that can reproduce asexually, and you are an individual plant in a new novel place, well, you are able to increase in numbers as a population if you are asexual. Then that it could be also good for settlement. And as I say, dispersal is not a single trait. It could be several traits, and it could be traits that are both that can be morphological. They could also be physiological, like related to metabolism. It can be behavioral, like I decide to lift my legs and fly away, or when do I gonna spawn as a fish. They can be also life history. That can related to fecundity, and it sounds a little bit funny, fecundity, because then you can think of clutch size for a bird. But if you think about plants, this is like the number of seeds, which is a very common trait for dispersal capacities. Okay, so then I am gonna go to the outcomes of my meta-analysis, in which I wanted to see all the factors that I told before, which ones are affecting how the relationship between dispersal and range size. So in general, I saw that the dispersal proxy we used was very relevant. Which ram a clade together, but in this case was not all the little clades that I showed before, but it's whether you are a freshwater animal or marine animal or terrestrial plants or terrestrial animals. The taxonomic breadth was important, the latitude and the taxonomic unit. And then you see the graph to the right. I can just describe more this. So then we see that terrestrial animals are doing a little bit, well, the effect on dispersal on range is less strong than for the rest of the groups. Also, studies on temperate regions, temperate and subtropical regions have stronger effects than in tropical and subtropical regions and along all the latitudes. We also see that actually studies using taxonomic levels that are at the level of division or phylum have very much like less strong effects than studies at the family or class level. When we go into terrestrial animals, we see that the dispersal proxy is very important, and the temperature regulation. In this case, endotherms seems to do a little bit less good. Well, the effect of dispersal is less strong than for ectotherms. And you can imagine that means that the dispersal might, how we measure dispersal doesn't affect range size. And I might have to do with something else, like maybe endotherms are able, despite of the dispersal capacities and airborne, when they arrive to a new place, they are able actually to tolerate new habitat because of the broad thermal tolerances. This could be one of the explanations. When you see all the different dispersal traits, you see all this variation. Then you see something that is very funny, is like for insects, everything is positive. Everything, all the traits show kind of positive effects, but the relative muscle mass shows actually negative effects. And then you might think having big muscles is good to disperse, but actually actually not, because they are costly, and then we have a trade-off, well, in this case. For birds, things related to the wing, like hindwing index, are traits that actually show a positive effect with range size, but body size, definitely not. For mammals, there is something going on with bats that you see a lot of variation, even if you just measure things related to wings. And in general, body size for mammals is a trait that is very complicated, and you see this huge variation. And this is because it looks like for mammals that are bigger than, I think, around 300 grams, the relationship is positive. But for mammals that are smaller than that, the relationship is negative, because there is a trade-off with fecundity. That is something, and therefore body size is a little bit tricky here. But for mammals, the maximum metabolic rate seems to be, well, affecting the range of the species. For reptiles, we don't see much, and then you see all this variation. In marine animals, no mammals, we see that all the different, the most important things are, well, several. There is, of course, the dispersal proxy, the latitude, the taxonomic breadth. And in the bottom, that you see all the different traits that people use. For fish, in general, the things are positive. Echinoderms, doesn't matter the trait, is very inconclusive what is going on, and there is not really an effect. But in general, for marine animals, the things are, the effects of dispersal on range are positive. And then you see here in the right what are the different effects depending on all these factors. Then we see that at the genus level studies, they show less strong effects. But in general, all dispersal always has a positive effect and on range. For plants, we only see two things that were important to explain the variation in the effect sizes, and this is the latitude and the taxonomic breadth. I anyway add the graph with all the different dispersal traits, and you see all this humongous variability. People that work with dispersal syndrome, well, is something that we have to think about. There is a lot of variation here. But okay, what we see in here that is interesting of the factors is that, as I thought before, studies at the family level or even at a genus level show stronger effects of dispersal on range size, but the studies at the division level do not show an effect. And this is because when people work with animals, maybe you just think of birds, mammals, fish, but but many of us might think of plants as plants, and you go to an area and let's measure the ranges of all the things together. And of course, it's all the traits that you are using for this might not really be meaningful for all of them, just to capture the dispersal, because they are different, biologically different. Okay, just to take, I want to wrap up with this, with some take-home messages. So I just showed that the relationships are mixed when we see the literature, but overall, the effect of dispersal is positive. We see that it's a large bias on the system, the studies, like the clades and the regions. There is definitely a lot of work to be done. I show that broader taxonomic delimitations obscure the relationship between dispersal and range size. And then I just put what would be the ideal level, like class or family. Definitely phylum or division is possibly not the way to go, because you might choose something that is not too different but not too similar. And of course, the choice of dispersal proxy is crucial. We really have to sit and think what would be the relevant traits, not only one, but maybe several, that can capture the complexity of dispersal. And I just want to take this opportunity to kind of promote a workshop that we are gonna do in winter this year, that has been funded by the Journal of Biogeography and Wiley from the small grants for global ecologia in biogeography. And this is together with Renske Onstein and Caroline Draxler, where we want to rethink what actually are dispersal-related traits. And we want to synthesize, of course, what dispersal is, how we measure dispersal, which traits we should consider to be good dispersal proxies, why, and of course we want to do this on all different taxonomic groups, not only the most widely known as birds, but we also want to include things that are really less studied, like soil organisms or planktonic life. So if you are interested on this, just drop me a line, or the information will be coming out in a couple of months. Just to finalize, of course, dispersal is important. And with this, I want to thank for the attention, and I just, happy to receive questions.
Well, thanks, Adriana, for your excellent talk. Many important things you avoided today. Sorry, you want to show another things?
No. That was the extra, just if they ask me questions.
Okay, nice. Well, now is open the space for the questions. Pessoal, agora vamos abrir então um espaço para as perguntas que a gente tenha para a Adriana. A gente pode escrever em português e a gente tenta traduzir para ela agora aqui, ou escrever diretamente em inglês para ela ajudar a responder. Então, enquanto o pessoal vai se animando aqui, fazer as perguntas, então uma das coisas que eu queria te perguntar, sorry, I speak in Portuguese now.
Just fine, just translate to me, thanks.
Yeah, well, I wait for the people sending the different questions with you, but I want to start the questions. And one question I have, I see you showed the different maps. But you show different methods. You use the different approach, the biological approach, the geographical approach, and evolutionary approach. I don't see nothing about the oceanography or the environment condition is important for drivers for the dispersion for many organisms. And what is your opinion about this one? Because, for example, in the ocean, many currents is very important for the transportation from in the larval state and many body size for different species. I don't know.
So of course, because we cannot go into much detail. If you want to do a meta-analysis on only marine systems, you might be able to do this, but because we wanted to do something more synthetic and include all the studies that have examined this question since the beginning of times, then you cannot really include these little details. So how we did was to include in this model, like in the meta-analysis, you just include a model in which let's say the outcome is the response variable is the effect size of the relationship, and then you put in the model, well, the different factors. In this case, I include the realm, and then I assume that in marine systems, the things are more connected, so it should be easier to disperse, therefore you might not see the relationship. But we actually saw that realm was not really important. That and was contrary to my hypothesis, because I was expecting that effect in marine systems was a little bit like less strong than in terrestrial systems. But then you actually know that you see that is actually quite positive. And in terrestrial systems, a little bit less strong, but it could be many other different things, like terrestrial systems has been also much more affected for human activities and these kind of things. So there are also many other factors that could just be playing a role.
Okay. Yeah. Oh, thanks. One point, please, speaking a little more slow.
Sorry. Yeah. I will try now.
Yeah. And well, another question is about the number of the studies you use it, because I see you have one bias very big when you try to find different factors. For example, I am expecting more restricting factors in dispersion in the freshwater systems in the Amazon River, for example. You have many canals and you have more endemic species, it's very restricting. But when you see it in the ocean, you have more dispersal and similar situation. You compare the Amazon forest or for another temporal forest, for example.
No, I definitely think that it should be different in freshwater systems. Like dispersal is very constrained to the how they call it, the basin. They're like, I mean, they are in a little basin and they don't go more than there. But the point is that there are not really many studies on freshwater systems. So when you just want to run a meta-analysis with like an n = 5, it doesn't really give you so it gives you a number, but it gives you a huge error there. So there is not much thing that you can conclude, and then you cannot also consider all the different factors. You cannot be sure of the factors because there is not enough sample. Like the sample size is very small. So if people started doing more studies on freshwater systems, it would be nice to see what happened. But it's indeed a pity. So I also wanted to show that is these groups that are really missing data. Yeah.
Nice. Well, another question I have when I see your presentation is about you see one different neutral process in different systems and different realms, but you after explain many traits is very important for the dispersal. And in your opinion, what is the more important trait or more important process you can describe? I know this is a big question.
Which is the most important trait for everything?
Yeah.
Do I have to say body size? Okay. Of course, body size can capture a lot of things, but as I show, for some organisms, definitely not. Like for birds, they fly. That doesn't matter their body. So I don't think we can really use like this is the answer for everything, body size. tree size over there, but I think we have to, for each group, let's say for each, that depends on the system, maybe for a single order, like for all fishes might be that body size is important. For plants, we might have to just not use a trait for all plants, but classify, okay, for plants that are actually maybe dispersed by no abiotic mediums, what will be the best traits? Plants that are dispersed by animals, what would be the best traits to use? And they're completely different.
Okay, nice. Well, this is my last question today, and this is more about the caramba, esqueci. Peraí. Well, I wait to the people make a question here. I remember again my question.
Yeah.
Pessoal, temos mais perguntas para a Adriana. Acho que a gente tem ainda 20 minutos para a gente conversar mais com ela, caso vocês tenham outras perguntas aqui, senão vai ficar um monólogo, só ela e eu aqui conversando. Não sei se vocês querem. Ah, tem uma pergunta aqui, ó. The assumption the species long range size may suffer dispersion limitation as far as assumption. I'm going to explain dispersion and migration colonization. Is the Bruno the question?
What can we? Maybe this is related to the 3 different phases of dispersal. I mean, we cannot assume that the species with small, I guess, we cannot assume that the species with the smaller ranges have dispersal limitations, because might be that you have a small range, but you are in an island. You can be very dispersive, but there is nothing around, that you are in a trap. But also might be also that you have large dispersal, but you're a new species, and new species start like with a small range. So there is a link that the smaller ranges are related to have less dispersal, but that doesn't, it's not a rule. And the other thing, I assume there are the 3 phases of dispersal, but I'm not sure if that is the case. I am not splitting dispersal in migration, colonization, and just trying to just conceive dispersal as something that is not only the movement. So it's not only just the movement from A to B, but it's also, it starts from before, like the decision to migrate. And there are several things that include that. And it finishes not only when you move, but you have to actually arrive. And in the very strict sense of dispersal, you have to just arrive and to kind of do this gene flow. And then, but for that, you have to colonize. So things that affect colonization should be also important. I don't know if that helped to answer the question. It's just, I don't want to split anything. I just want to just see that dispersal is more, it's more complex. It has several phases and it has several traits that are from different nature. And in addition, it can act at different life stages as an adult, as a juvenile, and it can, and different traits can affect different of the one or more of the dispersal stages. So it's yeah. I think the work on dispersal is still going on.
Yeah. Well, I remember my question now, and the question is about when you make it all the review for all these different articles, you found one relationship between the abundance or relative abundance and dispersion pattern?
Yeah, of course there is also the abundance things, but this is like another question. So of course there is, if you want to do, you're welcome, Bruno. Of course, if you want to do that for abundance, I guess that will be another review because there is a lot of data on that. I was collating also that data. And for instance, body size, in this case, I was working on dispersal. Of course, it came body size, but there is another body of literature only focusing on body size and range size, not assuming anything about dispersal. Mm-hmm. So there is, of course, well, a lot of data on it. But for abundance, there is also these mixed outcomes, for instance. I have done it. This is also, you just see both contrary kind of effects.
Yeah, because you mentioned about, well, I expected more the species has more range distance or more big distribution. Maybe is this species has more abundance, is more abundant
Yeah,
in general. But this is
depends.
only one's hypothesis I need to testing or you
No,
testing
no,
more.
it depends. What I have been thinking about that a lot, and what I think is not all, of course, abundance is important because the more abundant you are, then of course you have to occupy more space. You are many, you have to occupy the space, but then also there are more chances of disperse and more chances to arrive to new areas because you are more just by chance. But in addition, that doesn't just mean that because you're more abundant, you might have a large range, because you can be not very abundant, but you can have a very large dispersal. You might be only 2 individuals, but very large dispersal, and then you can occupy that. Well, you can just occupy a very large area, but let's say fragmented. So I guess it's also an interaction there with dispersal.
Okay.
So I have been doing this with some models, and this is what with neutral models, and this is what it shows.
Mm-hmm. Yeah. Well, this is more curious to do to when you try to explain for Bruno this question, your explanation about this one. I thinking about it. Is possible excluding all the paleoendemic species is very restricted. It's very all species and very restricted. But the more recently is have one small space range in general, or do
Which ones?
again? I try to explain. The more all species has more big.
The old.
Yeah, we have one geographical range in comparison for the species more recently is
That is,
more.
yeah, that is exactly the work we are working on right now. It is also in all different taxa. What I have found is that, well, there is a lot of variation for different groups. For reef fishes, I do see that. There is an issue, of course, when you just want to calculate the age of the species. Depends a lot on the phylogeny, how good they are. And you might tend, and depending when you don't include, when you ignore extinct species, then you overestimate ages of the species. So then this might be a little bit confounding, but we are working on that. And in general, we see that the age has a positive effect on range size, but for the moment is weak but positive. And of course, it's moderated by other things like dispersal of the continent where you live, or whether you live in an island or not. So yeah.
Well, nice answer, and thank you so much for this talk. I think this is very important. You mentioned topics today, and well, I don't know, the people have more questions. Pessoal, se tivesse mais perguntas, ainda temos 13 minutos a mais para conversar com a Adriana, senão eu vou dar por finalizado o nosso CEBIMário de hoje. E agradecer novamente ela estar participando aqui e trazer esse tópico interessante, recentemente aceito na revista Ecology Letters.
Yeah. Now you can, of course, always contact me, I don't know, in my email, if it's somewhere. Should be somewhere. And I'm happy to answer questions. Yeah. Or to discuss.
O contato da Adriana está diretamente na descrição do vídeo do YouTube, então qualquer que queira acessar, a ver o trabalho mais dela, vocês podem entrar lá e ver na descrição tanto dela quanto do paper recente que ela trouxe explicando hoje aqui para todos nós. Entonces, Adriana, muchas gracias nuevamente por traer este tópico. De nuevo estoy cambiando de idioma. Es chévere conocer más tu trabajo. Hace rato no hablamos y bueno, pues espero que sigas con ese tema así interesante y tenerte otra vez en un futuro aquí en este espacio para discutir más sobre tu trabajo.
Gracias de nuevo por la invitación. Thanks everybody for being here.
Ok, então, pessoal, eu espero vocês na próxima quinta-feira. A gente vai, então, continuar nossas palestras de pessoas internacionais. Vamos falar, então, de seagrass e vamos trazer o Dr. Jonathan Lepchecker falando um pouco sobre seagrass e escalas globais e demais. Então, até a próxima quinta-feira e obrigado a todos. Tchau.